Good morning, students. Today we are going to start with the second lecture of sexual reproduction in flowering plants. The major processes that we are going to study in this chapter: structure of flower, pre-fertilization events, fertilization and post-fertilization events. In the previous lesson, we have studied the structure of flower in detail, all the essential parts of the flower which are required for reproductive process. Then comes pre-fertilization events, because in sexual reproduction, what is important? It is fertilization. So before fertilization, we have to study the pre-fertilization events. That means we require gametes for fertilization and we have to prepare the gametes. In microsporogenesis, male gamete is produced and the female gamete is produced in megasporogenesis. Now coming to microsporogenesis, I had already done in the previous lesson. This is section of an anther showing 4 microsporangia. Okay, now this is one microsporangium, this is another microsporangium, and we have 4 microsporangia in one anther. Now each microsporangium is filled with sporogenous tissue, and the cells of sporogenous tissue undergo the process of meiosis to produce the microspore. And the process of meiosis takes place in 2 stages. You have studied these stages in class 11. Meiosis 1 results in the formation of haploid cells from the parent cell that is diploid, and after the division is completed, we get 4 haploid cells, and these are called microspores, which undergo the process of maturation to produce the pollen grains. Now, from the microsporangium, formation of the microspores which mature to produce pollen grains, this complete process is known as microsporogenesis. So we have completed the first process, that is in the pre-fertilization events we have completed microsporogenesis. This complete process I had discussed in the previous lesson. Now coming to the structure of a mature microspore, that is called pollen grain. This pollen grain has 2 cells, large cell which is filled with the cytoplasm to nourish the growing pollen grain. It is called vegetative cell, and the smaller one is called generative cell. It has 2 layers. The inner one is called intine and the outer one is called exine. The exine helps in protecting this pollen grain from harsh conditions like high temperatures and various conditions of desiccation, because of the presence of sporopollenin. This is 1 mark question. Name the hardest known substance present in the exine which helps the pollen grain to survive in the conditions of desiccation. So the answer is sporopollenin. Now this is 2 cell stage, and in 60% of angiosperms the pollen grains are released from the anther at 2 cell stage only, and in 40% of the angiosperms it is released at 3 cell stage, in which the generative cell nucleus divides into 2 before the formation of pollen tube. Now you remember, pollen tube will be formed on the stigma. Here when pollen grain fall, it will start forming pollen tube, And before the formation of pollen tube, the generative cell nucleus divides into two. So that will result in the formation of 3-cell stage. So in 40% cases, in angiosperms, 40%, it is released at 3-cell stage. Now this completes the microsporogenesis. Now we come to the second process, second pre-fertilization event, which is called megasporogenesis. In microsporogenesis, microspore was produced from microsporangium. But in megasporogenesis, megaspore will be produced from megasporangium. Now this comes the megasporogenesis process, that is formation of the female gametophyte. Now gynoecium, I told you in the previous lesson, this female reproductive part in the flower. If it has single pistil, it is called monocarpellary pistil. If it has many pistils, it is called multicarpellary pistil, and all the carpels, they may be united, as in case of poppy and hibiscus, it is called syncarpous multicarpellary ovary. If all of the carpels are separate, it is called multicarpellary pistil. If all of them are all the carpels are separate, it is called apocarpus. Example is Michelia. These examples are given in your NCERT and you can search out for more. Then this is the female gametophyte. This is gynoecium. If we undergo the transverse section of gynoecium, you can see it may have 2 compartments, 3 compartments, only 1, and this condition is also possible, free central position, in which the ovules which are developed, they are located in the center. So if we undergo the transverse section of the ovary, we can see the number of compartments. It may vary. Only 1 can be there, 2 can be there, 3 can be there, and these compartments are known as locules. So we can say it is multilocular ovary or bilocular. So this way we can name them. Now these in these locules at the placenta, this is placenta, at the placenta ovules may be attached. Okay, these ovules may be attached at the placenta, and the ovule is also known as megasporangia. Now coming to the structure of ovule. Ovule is also known as megasporangium, and one ovule has 2 parts. This is micropylar end and this is chelazal end. The ovule, which is also known as megasporangium. What was microsporangium? Microsporangium was a section of anther. This is section of anther in which we have 4 microsporangia. But here the megasporangium means ovule which is attached in the, you can say, because ultimately this ovule is going to be the, it will get converted into the seed on maturation. So the position of ovule can be seen within any fruit. For example, I am showing you pea. In pea plant, these are ovules. Okay, so how they are attached, this is called funicle. Now coming to the structure of this ovule, because ultimately this complete ovule is going to be converted into seed